Hello students and a warm welcome. I am Dr. Tripti Malikahuja. I am helping you in the preparation of various entrance examination for admission to the MSc Microbiology, Biotechnology, Biochemistry, Life Sciences, Bioinformatics and many more courses. In this video, you will get the question paper of BHU Applied Microbiology, which was conducted in the year 2018. In this year, if you're applying for the BHU Applied Microbiology course, then please note down that although it is conducted under the CUET examination, the exam is same, but here the question paper code is different. So if you are applying also to BHU, then you have to fill another paper code in your form. So go through the notification issued by NTA, so know where is the slot, which one is the paper code, so that you can also apply to the BHU. In the year 2018, there used to be a separate test, which was a paper-based test. There were the multiple choice questions in which there used to be the 4 options in which the correct option had to be circled by using a ball pen. Now this time there will be a computer based test, and the scheme of the question paper will be like that. There will be 100 questions. The first 25 questions will be on the general test basis, in which there will be questions from the English language, the verbal ability, logical reasoning and the quantitative aptitude. In the description box, I am giving some links of the books which can help you in the preparation of this general test. You can go through those books, and these books will help you in the preparation of the first 25 questions. The next 75 questions will be divided into the different categories, but for BHU, again note down that the 75 questions will only from the subject microbiology. The subject based test is having the questions only on the microbiology. So we are discussing this question paper. If you are new to this channel, then subscribe the channel and press the bell icon so that you can get the notifications of all the new videos. In the playlist, you will find that there are number of the videos which will help you in the preparation of specifically the CUET exam and other entrance examination. You can find the links in the description box below and in the I button here. So now let's start with this BHU question paper of 2018. This question paper had 120 questions. In this video, you will get the first 60 questions. In the second part, I will discuss the next 60 questions. So question number 1 to question number 60 in this video, question number 61 to question number 120 in the next video. As I am starting reading out these questions, I suggest you to take a pen and paper with you. As I am reading the question, you can write the correct option. If your option matches with the answer which I am giving you, then it's well and good. If it does not matches, I am trying to explain also, and still if you're having any doubt, you can write the comment section. I will answer to each and every comment. So question number first says, the mathematics of exponential growth in microorganism can be expressed as. There are 4 options, and don't worry, these options will seem like they are similar, but still they are different. Please pay attention and go through these options carefully. The first option, log n naught is equal to n log 2 plus log n. Second option, n log 2 is equal to log n plus log n naught. Third option, log n is equal to 2 log n naught. Fourth option, log n is equal to log n naught plus n log 2. So there are 4 options, and out of this you have to choose the correct option. So correct option is the option number 4. Now let's see what is this N0 and N. The N0 is the initial number of the bacterial cells present in a population, while N is the final number of the bacterial cells. Small n is the number of generations which this population is undergoing to producing the final number of the cells in the time period t. This equation can be followed when there is the exponential growth. Exponential growth means there is initially one cell, the one cell divides to give 2 cell, then 2 cell give to 4 rise to 4 cell, 4 to 8 and so on. So here the option 4th is correct. The log n is equal to log n0 plus n log 2. The second question, malolactic fermentation is carried out during the production of. The options are 1 beer, 2 wine, 3 biofuels, 4 cheese. So out of this the correct one is wine. Malolactic fermentation means in this fermentation there is production of malic acid as well as the lactic acid. In this fermentation the major product is although the alcohol, but these acids they are also produced. The malic acid gives a tart taste while the lactic acid gives a soft taste. So in wine there has to be a proper concentration of this malic acid and the lactic acid. Both these acids they are produced for the wine. Question number 13, a very simple question. Hope you will get the correct answer. Cyanotoxins are produced by 1 green algae, 2 blue green algae, 3 red algae, 4 yellow algae. So as you know that cyanotoxins they are produced by the blue green algae, also known as cyanobacteria. The question number 4, in a scheme of classification genetically related groups represent are. The option are 1 clone, 2 clade, 3 kingdom, 4 domain. The correct answer is clade. Clade constitutes of all the similar phylum. They are monophyletic. The question number 5 says, which one of the following is not found in phytoplasma cell membrane? Options are 1 protein, 2 lipids, 3 sterols, 4 fatty acids. The correct answer is the 3 sterols. Sterols are found in the cell membrane of fungus. In the case of phytoplasma, the cell membrane is triple layered. Question number 6. Clinically useful aminoglycosides include. The options are 1 penicillin, 2 cephalosporin, 3 streptomycin and 4 erythromycin. Here the correct option is the option number 3, which is streptomycin. The question number 7. Tipicolonic acid is formed mainly in which of the following bacterial structures? Options 1 flagella, 2 sex pilus, 3 capsule, 4 endospore. Here the correct one is the 4 endospore. Question number 8. E. coli genomic DNA has approximately how many base pair? The options are 1 4.5, 1.8 MB, 2.1 MB, 8.5 MB. 1 MB means 10 raised to power 16. That means 1 million bases. For E. coli, the number of base pairs is 4.5 MB. Question number 9. Again a very simple question. Vaccination was developed by 1 Stanley Brusner, 2 Edward Jenner, 3 Paul Ehrlich, 4 Robert Koch. The correct answer is Edward Jenner. The first vaccine was the smallpox vaccine. Question number 10. Genomic concatenameric DNA is formed during the replication of 1 bacteriophage, 2 bacteria, 3 yeast, 4 plant virus. The correct answer is the 1 bacteriophage. Next question, number 11. Bacteroides surrounded by a plant cytoplasmic membrane form structures called as infection thread, symbiosome, nod factors, root nodules. The correct answer is the option number 1, which is the infection threat. It is seen in the case of Rhizobium and other symbiotic bacterium. The knot factors, they are secreted by the bacteria, and the symbiotic exopolysaccharide form structures which are called the infection threat. The question number 12. Which one of the following has been used to enrich rice paddies with fixed nitrogen? 1. Azolla, 2. Rhizobium, 3. Streptomyces, 4. Agrobacterium. The correct one is the option 1, which is Azolla. Azolla, these are the freshwater fern which form symbiotic nitrogen fixation via the cyanobacteria, and they enrich the soil with nitrogen. Question number 13. The cos sites of bacteriophage lambda is made up of how many nucleotides? The options are 8, 12, 16 and 20. The correct one is the option number 2, which is 12. The next question number 14. Which one of the following is not a mutagen? Or the question is very simple, but still it is tricky because it is written not a mutagen. First of all, sometimes we read just only mutagen and we tick the first option. But be careful, read the question carefully. It is saying not a mutagen. So the options are ethadium bromide, X-ray, transposones, salicylic acid. So out of this, there is one which is not a mutagen, and that is the 3 transposones. The question number 15. The lac repressor functions as 1 monomer, 2 dimer, 3 trimer, 4 tetramer. So here the correct answer is the 4, which is tetramer. Lac repressor mediates the formation of DNA loops by simultaneous interaction with single protein. Then it forms a tetramer with the DNA binding sites. Question number 16. Now it is written aoxygenic. Okay, so this is somewhat different. It should be, I think, it should be the oxygenic. Okay, phototroph prochlorophyte contains, the options are phycobilins and chlorophyll B. Then there are the chlorophylls A and B and no phycobilins. Option 3, phycobilins and chlorophylls A and B. 4, phycobilins and no chlorophylls. So out of this, the correct option is the option number 2. So it is a oxygenic phototrope. That means there are the oxygenic phototrophs. These are also called prochlorophytes. These are oxygenic photosynthetic cyanobacteria which are known for their photosynthetic pigments, and they lack phycobilins, but they have unusual chlorophyll. So they have the chlorophyll A and B but not phycobilins. The question number 17 says, which of the following is a MOFE protein? MO is for molybdenum, Fe is for the iron. The options are dinitrogenase. Number 2 option is dinitrogenase reductase. Number 3, 1,3-beta glucanase. 4 is DNA polymerase. The correct one is the option number 1, dinitrogenase. The question number 18 says, taking up DNA by transformation is an inherited property of a bacterium. This is due to presence of number 1, COM protein genes, number 2, NIF genes, number 3, trip operon, number 4 is the siRNA. So out of this, the first one option is correct. The first option, which is the COM protein genes. COM protein genes are present in an operon which are responsible for their competence of the bacteria. Question number 19. A mutant with a growth requirement for a specific nutrient is known as number 1, autotroph, number 2, auxotroph, number 3, heterotroph, number 4 is the phototroph. So here the correct option is the option number 2, which is the auxotroph. The question number 20. Some of the metabolic plasmids of bacteria carry genes for enzymes that direct the formation of sex pili, destruction and modification of antibiotics, degradation of aromatic compounds. Fourth is the killing of other bacteria. So out of this, the option number 2 is correct, the destruction and modification of antibiotics. Question number 21. Chemoautotrophic bacteria derive energy for their physiological needs by 1. Oxidizing organic compounds, 2. Reducing nitrogen, 3. Absorbing solar energy, 4. Oxidizing inorganic chemicals. The correct answer is the 4. Oxidizing inorganic chemicals. These chemoautotropic bacteria, they oxidize inorganic substances such as ammonia, molecular hydrogen, sulfur, hydrogen sulfide and ferrous ions. The question number 22, a very simple direct question. Red rust of tea disease is caused by a member of 1. Algae, 2. Bacteria, 3. Plant viruses, 4. Fungi. So for this question, the correct answer is 1. Algae. The red rust of tea is a very unique disease which is caused by an algae. The name of the algae is Cephalaris parasiticus. The question number 23, again a very simple question. This question says the term cistron was given by 1. Mueller, 2. Sutton, 3. Benzer, 4. Nirenberg. So out of this, the correct answer is the option number 3, which is Benzer. The term cistron was given by Benzer. The cistron means the genes which exhibit a specific behavior in cis-trans test. Question number 24. The famous microbiologist who disapproved the theory of spontaneous generation was 1. Carl Woese, 2. Martin Beijerinck, 3. Louis Pasteur, 4. Stanley Miller. So the correct one is Louis Pasteur. Hope you remember the experiments, the Swan experiments which he carried to disapprove the theory of spontaneous generation. The next question, number 25. The mode of nutrition of methanogenic bacteria is 1. Chemoautotrophic, 2. Photoheterotrophic, 3. Chemoorganotrophic, 4. Auxotrophic. So the correct option is there, chemoautotrophic. Chemoautotrophs, they obtain the energy from electrons which are found in the hydrogen gas and produce methane and other organic compounds. Question number 26, a different question. Select the mismatch. So here there are 4 pairs and 1 pair is mismatched. You have to identify which pair is mismatched. So option number 1, phycoerythrin pigment, non-photosynthetic. Option 2, heterocyst anaerobic cell. Option 3, bacterial endospores, heat resistant cells. Option 4, ammonium nitrogen, the electron donor. So out of this, the mismatched is the first one, which says phycoerythrin and the non-photosynthetic. Actually, the phycoerythrin, these are the accessory photosynthetic pigments which are present in the photosynthetic bacteria. So instead of non-photosynthetic, if it should be the photosynthetic, then it should be correct. But here there is mismatching, so we have to tick the option number first as the correct option. The question number 27. The association and dissociation of ribosomal subunits are dependent on the concentrations of option 1, magnesium ion, number 2, calcium ions, number 3, sodium ions, number 4 is the manganese ions. So here the correct one is the option 1, which is the magnesium ions. Question number 28. if a bacterial cell divides in every 20 minutes, how many bacterial cells will be formed in 2 hours? So options are 16, 24, 64 and 32. So out of this, the correct one is the option number 3, which is the 64. One bacterial cell gives rise to 2. After 2, 4 are formed. In next division, 8 are formed. In next 16, from 16 to 32, and then 32 to 64. And if you note down the period in between all of this division, the total time comes to be the 2 hours. That means the 120 minutes. A very simple question, but it requires some calculations. So the question number 29, the main biological function of naturally occurring bacteria associated with the gold and copper mines is: 1. To oxidize, reduce sulfur and form sulfuric acid. Number 2, to convert ammonia to nitrate. Number 3, to oxidize ferric to ferrous. The ferrous to ferric. Then the number 4, to fix nitrogen. So out of this, the correct option is the option number 1, which is to oxidize reduced sulfur and form sulfuric acid. In the case of the mines, the gold and copper mines, the bacteria which is utilized is the acidiobacillus ferrooxidans. So this bacteria is a bacillus. The name is, again I am repeating, acidiobacillus ferrooxidans. It is used for bioleaching. Bioleaching means the metasolubilization, which helps in solubilizing the sulfides matrix of the gold deposits, making gold more reachable so that it can be leached out. Question number 30. When an old bacterial culture is transferred to the fresh basal medium, the lag growth phase will be, okay? The lag is a part of the growth curve. The options are prolonged, absent, reduced, without any change. The answer is, it is without any change. A bacterium has to adjust to the fresh medium, so there will always be a lag period. A lag period is a period when the bacterium is minimally dividing. So the growth curve is in this form, the S-shaped, and the lag period is the period in which there is very less division of the bacterial cells. The bacterial cells, they are hardly dividing. Question number 31 says, when mutation occurs due to the substitution of a pyrimidine base by a purine base, it is called transition, transgenic, transformation and transversion. It is called transversion. So option number 4 is correct. Question number 32. The superbug, a transgenic bacterial strain, was created to clean the pollution caused by 1 pesticide, 2 eutrophication, 3 heavy metals, 4 petroleum hydrocarbons. So out of this, the correct option is the option number 4, which is petroleum hydrocarbons. This was the superbug created by the Anand Mohan Chakravarti to clean oil spills. Question number 33. Again, it involves number of some figures, so you have to listen it carefully. How many quanta of light energy are required for the use of 4 positive equivalents with the production of 4 reducing equivalents necessary for the evolution of 1 molecule of oxygen from 2 molecules of H2O, that means water? The options are 8, 12, 6 and 4. Out of this, the correct one is the option number 1, which is the 8. The 8 quanta, these are required so that the number of oxygen molecules which are released per photon or from the quantum of light. This is known as the quantum yield. So there is a requirement of at least 8, sometimes it can be the 10 quanta also. So this quanta of light, they are required to evolve one molecule of oxygen, or in this process one molecule of carbon dioxide is consumed. So one molecule of oxygen is produced while one molecule of carbon dioxide is consumed. So question number 34. The entire network of cell cytoplasm of plant cells interconnected by plasmodesmata is referred as apoplast, spheroplast, symplast or protoplast. Out of this, the correct one is the option number 3, which is the symplast. In plant cells, plasmodesmata establish an interconnected cytoplasm called symplast through the metabolites, which are proteins and RNAs, which are exchanged between the cells, tissues and organs. Question number 35. Which of the following is primary transporter? The options are antiporter, symporter, uniporter, ABC transporter. So out of this, the correct option is the option number 4, which is the ABC transporters. These ABC transporters, they mediate import and export processes, which acts as the ion channels or the regulators. Question number 36. Cyanobacteria differ from purple and green phototrophic bacteria because they, number 1, show oxygenic photosynthesis, use hydrogen sulfide as electron donor. Option 3, have a membrane and closed nucleus. Option 4, don't require light. So out of this, the correct option is the option number 1. They show oxygenic photosynthesis. There are 2 linked photosystems in which the hydrogen and there is a water and there is an electron donor. Question number 37. Which of the following is not a sink in plants? Now again a simple question, but still you have to think it over. The options are flower bud, the option 2 developing fruit, the option 3 photosynthetically active leaf, the option 4 a storage organ of the plant. The first of all, out of this, the sink which is there, the sink is the option 3, which is the photosynthetically active leaf. Now listen carefully, what is a source, what is a sink? Source is the potential capacity for the photosynthesis. That means where there is the photosynthesis is there, and the sink means in which there is the utility, where there is the consumption of the photosynthetic products. So sink can be all the sites, it can be a flower bud, a fruit, a storage organ, but source is always where there is the photosynthesis. The option number 38. Zygote is characterized by 1 synapses crossing over, tetrad formation, 2 synapses bivalent crossing over, 3 recombination, nodule synapses and bivalents, 4 bivalent synapses and tetrad formation. So out of this, the correct is the option number 1, in which the sequence is proper sequence, the synapses crossing over and the tetrad formation. Coming to question number 39. Which of the following is arginine rich? The options are H1, H2A, H2B and H3. So out of this, the correct one is the option number 4, which is the H3. H stands for the histone. The histone protein 3 is rich in arginine. In addition to the H3, H4 is also rich in arginine. while the H2A and the H2B histones, they are rich in lysine. So you can see there are a lot of questions from the cell biology. So cell biology is important for the entrance examination. Another question from the cell biology is question number 14. Cell cycle is regulated by the master control molecules known as 1 transferases, 2 lipases, 3 kinases, 4 dehydrogenases. So here the correct one is the option number 3, which is kinases. The question number 41, which of the following is a microfilament? The options are 1 keratin, 2 actin, 3 desmin and the 4 tubulin. The correct option is the option number 2, which is the actin. So actin are the protein microfilaments which are found in the cytoplasm of the eukaryotic cell, which had to form the cytoskeleton. Question number 42. Which of the following is hemizygous? The option 1, male mice. Option 2, male Drosophila. Option 3, female Drosophila. Option 4, male plant of Melendrium. Now note down, the question says hemizygous. So when we say the hemizygous, the males are always hemizygous since they contain only one X chromosome and the other one, which is the Y. That means only half of the alleles are normally present for a diploid individuals. So if we consider these 4 options which are given here, then out of these 4, the first option, male mice, obviously male mice is XY, so it is hemizygous. Male Drosophila is also hemizygous. Then the male plant of the Melendrium is also hemizygous. So the which is not hemizygous is the female Drosophila. So this question according to me is not correct. The question which is asked here, the question which of the following is hemizygous is not correct. The question should be which of the following is not hemizygous. So there should be a word added here, not hemizygous. Then the option could be the option number 3, female Drosophila. So there could be an objection to this question. The question is incorrect. The question number 43. Which of the following combination is true as proponents of synthetic theory of evolution? The options are Dobbs-Henke, Fischer, Lamarck, Haldane, Ernest Mayr. Option 2, Dobbs-Henke, Fischer, Darwin, Haldane, Ernest Mayr. Option 3, again Dobbs-Henke, Fischer, Hugo de Vries, Haldane, Ernest Mayr. Option 4, Dobbs-Henke, Fischer, Haldane, Ernest Mayr, and Sievelwright and Stebbins. So out of this, the correct option is the option number 4. There is a new Darwinism theory according to which the genetic changes which occur in a population, they lead to the formation of new species. So that is also a proponent of the synthetic theory of the evolution given by Dobbs-Henke, Fischer, Haldane, Ernest Mayr, Wright and Stebbins. Question number 44, a very simple question. The linkage map is also referred to as 1 chromosome map, 2 physical map, 3 restriction map, 4 genetic map. So here I hope you have guessed the correct answer, and yes, the correct one is the option number 4, which is the genetic map. Question number 45. In glycolysis, fructose 6-phosphate is transformed to fructose 1,6-diphosphate by the enzyme. The options are 1 hexokinase, 2 phosphohexoisomerase, 3 phosphofructokinase, 4 phosphotriose isomerase. So here the correct one is the option number 3, the enzyme phosphofructokinase. In this step, the second molecule of ATP is added. Now the question number 46, very simple question. Chitin is a 1 polypeptide, 2 polysaccharide, 3 polyphosphate, 4 lipid. So here the correct option number 2, which is polysaccharide. Question number 47, which of the following enzyme is responsible for DNA chain elongation? The options are 1 DNA polymerase 1, 2 DNA polymerase 2, 3 DNA polymerase 3, 4 is the RNA polymerase. So out of this, the correct one is the option number 4, which is the RNA polymerase. The question number 48. Agrostil electrophoresis is used for separating 1 proteins, 2 nucleic acid, 3 lipids, 4 carbohydrates. So here the correct one is the option number 2, which is the nucleic acids. Question number 49. Which the following element is responsible for evolving oxygen in plant through splitting of water by changing its oxidation states? The options are number 1 Fe, number 2 magnesium, number 3 manganese, and min number 4 is the copper. So here the correct one is the option number 3, which is the manganese. Number 50 question. Psammophytes grow on 1 stone, 2 saline land, 3 sand, 4 is the marshy lands. Here the correct one is the option which is the sand. The psammophytes, these are the plants that thrive in the shifting sands, primarily in deserts. These are small shrubs, trees, and they form the adventitious roots. Now question number 51. Stone leprosy is caused by 1 Mycobacterium leprae, 2 lightning, 3 acid rain, 4 dust on the sand. Out of this, the correct option is the option number 3, which is the acid rain. It's a very different question. The question is saying leprosy. So when we think of leprosy, it comes that it is a bacterial disease caused by the Mycobacterium leprae. So it would be because of this Mycobacterium leprae. But here, this stone leprosy, actually it is because of the acid rain. The acid rain causes substantial damage to the buildings which are made up of marble and limestone. When there is the attack of acid rain on the marbles, then it is called stone leprosy. Question number 52. The site of glycosidation of lipids and proteins to produce glycolipids and glycoproteins in the cells is 1 mitochondria, 2 chloroplast, 3 Golgi complex, 4 lysosomes. So here the correct option is the option number 3, which is the Golgi complex. Question number 53, a very direct and simple question. 1 gene, 1 enzyme hypothesis was given by 1 Beadle and Tatum, 2 Jacob and Monod, 3 Watson and Crick, 4 Luria and Delbruck. So I hope you have guessed the correct answer. So the correct one is the option number 1, Beadle and Tatum. Question number 54. Clathrin coated vesicles are meant for 1 extracellular traffic, 2 intercellular traffic, 3 coating vacuole, 4 protein synthesis. So here the correct one is the option number 2, which is intracellular traffic. The intercellular traffic means transport of proteins from organelles to the different organelles. The transport between one organelle to the other organelle. Question number 55. Which one of the following are terminator codon? The options are UA, UAG, UGA, AUG, UAG, UGA, UAC, AUG, UAG, AUG, ACG, GAG. The correct option is the option number 1, which is UAA, UAG and UGA. All these are called the stop codons, nonsense codons, or these are also called the amber codons. Next question, number 56. The starting transfer RNA of prokaryotes is loaded with. The options are valine, methionine, tryptophan, formylated methionine. So here the correct option is the option number 4, which is formylated methionine. The initiator transfer tRNA interacts, the initial transfer RNA interacts with the start codon AUG of the messenger RNA and carries the formylated methionine, which is also called FMET. Question number 57. Which of the following contains hydrolytic enzymes? The number 1 dictyosome, number 2 peroxisomes, number 3 lysosomes, number 4 carboxysomes. So here the very simple question, I hope you have written the correct answer on the paper. Yes, the option number 3 lysosomes, it is the correct answer. Lysosomes, they are also called suicidal bags of the cell. Question number 58. Km, Michaelis-Menten constant, is defined as. The option 1, substrate concentration at which all of the enzyme molecules are forming ES complex, enzyme substrate complex. The substrate concentration at which 3/4 of the enzyme molecules are forming ES complex. The option 3, the substrate concentration at which half of the enzyme molecules are forming the ES complex. Option 4, the substrate concentration at which 1/3 of the enzyme molecules are forming the ES complex. So here the option number 3 is correct. It is that substrate concentration at which half of the enzyme molecules are forming the ES complex. Now moving to question number 59. The study of genetic material recovered directly from the environmental samples is known as 1 metagenomics, 2 proteomics, 3 genomics, 4 metabolomics. So it is the option number 1, which is metagenomics. So here genetic material is recovered from environmental samples and it is directly processed for the extraction of DNA and the sequencing and other techniques. Question number 16. MAB stands for. The option are 1, Man and Biology, number 2, Man and Biosphere Program, number 3, Map and Biology, number 4 is Management and Biosphere. So here the correct one is option number 2, the Man and Biosphere Program. It is the international biological program of UNESCO, started in the year 1971, but in India it was started in 1980. So these were the first 60 questions. The next 61 to the 120 questions, the next 60 questions will be answered in the part 2. So you will get the link soon. Stay connected, stay tuned. Best wishes for your exam.